Chapter 6 Anatomy of Flowering Plants. Study of internal structure of plant is called anatomy. Anatomy. Internal structure does not show adaptation to diverse environments. False. In tissues, cells have different origins but common functions. False. Have common origins and may have common function. False. Have common origins and may have common function. False. Have common origins and may have common function. Meristematic tissues. Specialized regions of active cell division are called meristems. Greek word dash means divided. Meristos. Apical meristems that produce primary tissue are called apical meristems. Identify the tissues shown in diagram 1, label A to G. Diagram 1 is root apical meristem. A is central cylinder, B is cortex, C is protoderm, D initials of central cylinder and cortex. E is initials of root cap, F is root cap, and G is root apical meristem. Diagram 1 is root apical meristem. A central cylinder, B cortex, C protoderm, D initials of central cylinder and cortex, E initials of root cap, F root cap, G root apical meristem. Diagram 1 is root apical meristem. A is central cylinder, B cortex, C protoderm, D initials of central cylinder and cortex, E initials of root cap, F root cap, and G root apical meristem. Identify the tissue shown in diagram 2, label A to E. Diagram 2 is root apical meristem. A is leaf primordium, shoot apical meristem, and A is leaf primordium, B is shoot apical, C is meristematic zone, and D is axillary bud, and E is differentiating vascular tissue. Diagram 2 is shoot apical meristem. A is leaf primordium, B is shoot apical, and C is meristematic zone, D is axillary bud, and E is differentiating vascular tissue. Shoot apical meristem. A is leaf primordium, B is shoot apical, C is meristematic zone, D axillary bud, and E is differentiating vascular tissue. Some left behind cells from shoot apical meristems constitute the axillary bud. Axillary buds are present at dash and are capable of forming a axil of leaf or branch or flower. Axil of leaf and are capable of forming a branch or flower. Example of primary meristems: apical and intercalary meristems. Define intercalary meristem. Meristem which occur between mature tissues. Intercalary meristems occur in grasses. Secondary meristems, also called dash meristem or dash meristems. Lateral meristems are cylindrical meristems. Example of primary meristems: examples of lateral meristems: interfascicular cambium, car cambium, and fascicular vascular cambium or intrafascicular cambium. Interfascicular cambium, car cambium, and fascicular vascular cambium or intrafascicular cambium. Interfascicular cambium, car cambium, and fascicular vascular cambium or intrafascicular cambium. Intrafascicular cambium, also called fascicular vascular cambium. Dermal tissues, ground tissues, and vascular tissues are formed by apical meristem. True. Permanent tissues. Simple tissues. Dash form the major component within organs. Parenchyma. Parenchyma shapes. Variable. variable. Parenchyma have thick or thin walls made up of thin. Parenchyma cannot be closed. False. It can be closely packed or have small intercellular spaces. False. It can be closely packed or have small intercellular spaces. False. It can be closely packed or have small intercellular spaces. Identify the simple tissues shown in diagram 3, X, Y and Z. Label A to J. Diagram 3, X is parenchyma, X is parenchyma, A intercellular space, Y is collenchyma, B thickened corners, C protoplasm, D vacuole, E cell wall, and Z is sclerenchyma, F lumen, G thick cell wall, and H pits, I is lumen and J thick cell wall. Diagram X is parenchyma, A intercellular space, Y collenchyma, B thickened corners, C protoplasm, D vacuole, E cell wall, Z sclerenchyma, F lumen, G thick cell wall, H pits, I lumen, J thick cell wall. Parenchyma functions: photosynthesis, storage, secretion. Photosynthesis, storage, secretion. Photosynthesis, storage, secretion. In most monocot or dicot plants, collenchyma occurs in layers below dash. In dicot, below epidermis. Dicot, below epidermis. In dicot, below epidermis. Cells in collenchyma are thickened at the corners of the deposition of cellulose, hemicellulose and pectin. Cellulose, hemicellulose and pectin. Cellulose, hemicellulose and pectin. Intercellular spaces may be present in collenchyma. False. Totally absent. False. Totally absent. False. Totally absent. Sclerenchyma provides support to petiole of leaf and young stem. False. Collenchyma does provide the support to petiole of leaf and young stem. False. Collenchyma does. False. Collenchyma does. Sclerenchyma usually contains protoplast. False. They are dead. They do not contain the protoplast or protoplasms. False. They are dead. False. They are dead. Sclerenchyma consists of long or short narrow cells with thick or thin cell wall. Long thick cell wall. Long thick cell wall. Cell wall in sclerenchyma is dashified, lignified. Name the two types of sclerenchyma: fibers and sclerates. Sclerates are found in fruit wall of nuts, pulp of fruits like guava, pear and sapota, seed coat of legumes, leaves of tea. Fruit wall of nuts, pulp of fruits like guava, pear and sapota, seed coat of legumes and leaves of tea. Fruit wall of nuts, pulp of fruits like guava, pear and sapota, seed coat of legumes, leaves of tea. Collenchyma provide mechanical support to petiole of leaf and young stem. Sclerenchyma provide mechanical support to organs. Complex tissues. Identify the complex tissues shown in diagram 4, X and Y. Label A to F. Diagram 4, X is xylem, A is trachea, B vessels, Y is phloem, C pore, D C tube element, E phloem parenchyma and F companion cell. X is xylem, A trachea, B vessels, Y is phloem, C pore, D C tube element, E phloem parenchyma, F companion cell. 4 elements of xylem are tracheates, vessels, xylem parenchyma, xylem fibers. Tracheates, vessels, xylem parenchyma, xylem fibers. Tracheates, vessels, xylem parenchyma, xylem fibers. Xylem provides mechanical strength to the plant. True. Main water transporting elements in flowering plants are vessels. Living element of xylem is xylem parenchyma. xylem parenchyma. Dash lack vessels in their xylem. Gymnosperms. Tracheids have thin and lignified walls with tapering ends. False. Vessels also have tapering ends. False. Vessels is made of many cells called vessel members. Vessel members have lignified walls. True. The vessel cells are also devoid of protoplasm. True. Vessel members are not interconnected through perforations. False. Characteristic feature of angiosperm is presence of dash in xylem. Vessels. Xylem fibers have obliterated central lumens. True. Xylem fibers are always septate. False. Xylem parenchyma is thin walled and the cell wall is made up of cellulose. True. Xylem parenchyma store food in the form of dash or dash and other substance like dash, starch or fat, or other substance like tannins. Starch or fat, tannins. Radial conduction of water takes place by dash cells. Ray parenchymatose. Primary xylem types: protoxylem and metaxylem. First formed primary xylem is called dash and later formed is called dash. First formed is protoxylem and later formed is metaxylem. First formed is protoxylem and later formed is metaxylem. In stems, protoxylem lies towards the dash and the metaxylem lies towards dash. In stems towards center, that is protoxylem towards center and metaxylem lies towards the periphery. Protoxylem towards center and metaxylem towards periphery. Protoxylem towards center and metaxylem towards periphery. Periphery means outer side. In stem, xylem arrangement is endark or exarch. In stem, endark. In root, xylem arrangement is endark or exarch. Phloem composed of sieve tube elements, companion cells, phloem parenchyma and phloem fibers. Sieve tube elements, companion cells, phloem parenchyma and phloem fibers. Sieve tube elements, companion cells, phloem parenchyma and phloem fibers. In gymnosperms, dash and dash components of phloem are replaced by dash and dash respectively. Sieve tubes, companion cells by sieve cell and albuminous cells. Sieve tubes and companion cells are replaced by sieve cell and albuminous cells. Sieve tubes and companion cells are replaced by sieve cell and albuminous cells. Sieve plates are found in end or between of sieve tube. The end wall of dash are perforated in a dash manner to form dash. The end wall of sieve tube are perforated in a sieve-like manner to form sieve plates. Sieve tube, sieve-like, sieve plates. Sieve tube, sieve-like, sieve plates. Sieve elements contain a nucleus. False. Dash are specialized parenchyma or collenchyma cells which are associated with sieve tube. Companion cells. Sieve tube and companion cells are connected by pit fields. Companion cells help in maintaining the dash and sieve tubes. Pressure gradient. Phloem parenchyma stores dash and other substance like dash. Phloem parenchyma stores food material and other substance like resins, latex, mucilage. Food material, resins, latex, mucilage. Phloem parenchyma are elongated, tapering and cylindrical cells. True. Phloem parenchyma is absent in most of the monocot or dicot. monocot. Phloem fibers are also called bast fibers. Bast fibers. Bast fibers. Phloem fibers are made up of dash chyma, sclerenchyma, sclerenchyma, sclerenchyma. Phloem fibers are generally present in primary phloem. False. Absent in primary but present in secondary phloem. False. Absent in primary but present in secondary phloem. False. Absent in primary but present in secondary phloem. Phloem fibers of dash 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 are used commercially. Flax, hemp and jute. Flax, hemp and jute. Flax, hemp, jute. Metaphyloem have bigger sieve tube than protophyloem. True. 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 The tissue system 74. On the basis of structure and location, the three types of tissue systems are ground tissue system, epidermal tissue system, vascular tissue system. Ground tissue system, epidermal tissue system, vascular tissue system. Ground tissue system, epidermal tissue system, vascular tissue system. Epidermal tissue system comprises epidermal cells, stomata, epidermal appendages, trichomes and hairs. Epidermal cells, stomata, epidermal appendages, that is trichomes and hairs. Epidermal cells, stomata, epidermal appendages, that is trichomes and hairs. Epidermis have compactly arranged cells. True. 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 Epidermis is usually single layered. True. 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 Epidermis cells are sclerenchymatous. False. Parenchymatous. 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 Epidermis have large or small amount of cytoplasm lining the cell wall and a large or small vacuole. Small amount of cytoplasm and a large vacuole. Small amount of cytoplasm and a large vacuole. Small amount of cytoplasm and a large vacuole. Epidermis have. Cuticle prevents loss of water. 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 Cuticle is absent in roots. 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 Dash regulate process of transpiration and gaseous exchange. Stomata. 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 Identify the labelings in the diagram of stomata shown in diagram 5. Diagram 5 is epidermal cells. A is epidermal cells, B subsidiary cells, C chloroplast, D guard cells and E stomatal pore. A epidermal cells, B subsidiary cells, C chloroplast, D guard cells and E stomatal pore. A epidermal cells, B subsidiary cells, C chloroplast, D guard cells and E stomatal pore. In dash, guard cells are dumbbell shaped. In monocots, guard cells are dumbbell shaped. In monocots. The outer wall of guard cell is thick while in the inner wall is thin. False. Outer wall is thin and inner wall is thick. False. Outer wall is thin and inner wall is thick. False. Outer wall is thin and inner wall is thick. Guard cells don't have chloroplast. False. They have chloroplast. False. They have chloroplast. False. They have chloroplast. Dash cells regulate opening and closing of stomata. Guard cells. Guard cells. Guard cells. Few epidermal cells in the vicinity of the guard cells become specialized in the shape and size and are known as subsidiary cells. Subsidiary cells. Subsidiary cells. Stomatal apparatus components are guard cell, subsidiary cell, stomatal aperture. Guard cell, subsidiary cell, stomatal aperture. Guard cell, subsidiary cell and stomatal aperture. Root hairs are unicellular or multicellular. Unicellular. 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 Trichomes are usually uni or multicellular. 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 Trichomes help in preventing water loss due to transpiration. 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 All tissues except epidermis and vascular bundles is called ground tissue. Ground tissue. Ground tissue. Ground tissue doesn't contain sclerenchyma. False. It contains all parent, colon and sclerenchyma. Ground tissue contains all parent, colon and sclerenchyma. False. It contains all parenchyma, collenchyma and sclerenchyma. That is ground tissue have all these three. In leaves, ground tissue is called mesophyll. 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 Identify the types of vascular bundles shown in diagram 6 A to G. Diagram 6, X is radial, X, A xylem, B phloem, Y is conjoint closed, C phloem, D xylem, Z conjoint. X is radial, A xylem, B phloem, Y conjoint closed, C is phloem, and D is xylem, Z conjoint. Open, E phloem, F cambium, G xylem. Z is conjoint open, E phloem, F cambium, and G xylem. In monocot or dicot stem, cambium is present between phloem and xylem. Dicot. What are open vascular bundles? Vascular bundles which, because of the presence of cambium, possess the ability to form secondary xylem and phloem tissues. Vascular bundles which, because of the presence of cambium, possesses the ability to form secondary xylem and phloem tissues. Vascular bundles which, because of the presence of cambium, possess the ability to form secondary xylem and phloem tissues. Radial arrangement happens in dash manner along different dash and is seen in stem or root. Alternate manner along different radii and is seen in root. Alternate manner along different radii and is seen in root. Alternate manner along different radii and is seen in root. In dash type of vascular bundles, the xylem and phloem are jointly situated along same or different radii. In conjoint type of vascular bundles, the xylem and phloem are jointly situated along same or different radii. Yes. In conjoint in same radii. Yes, in same radii. In dash type of in conjoint type of vascular bundles, the xylem and phloem are jointly situated along the same radii. Conjoint same radii. Conjoint vascular bundles have phloem located only on the inner side of xylem. Falls on outer side of xylem. Anatomy of dicot and monocot plants. Identify the tissues given in diagram 7 A to I. Monocot. Diagram 7 is monocot root. A is root hair, B epidermis, C cortex, D endodermis, E pericycle, F phloem, G protoxylem, H pith, I metaxylem. Monocot root. A is root hair, B epidermis, C cortex, D endodermis, E pericycle, F phloem, G protoxylem, H pith, and I is metaxylem. Monocot root. A root hair, B epidermis, C cortex, D endodermis, E pericycle, F phloem, G protoxylem, and H pith, I metaxylem. Identify the tissue given in diagram 8. Label A to I. Dicot root. A root hair, B epidermis, C cortex, D endodermis, E pericycle, F protoxylem, G metaxylem, H pith, I phloem. Dicot root. A is root hair, B epidermis, C cortex, D endodermis, E pericycle, F protoxylem, G metaxylem, H pith, I phloem. Dicot root. A root hair, B epidermis, C cortex, D endodermis, E pericycle, F protoxylem, G metaxylem, H pith, and I phloem. Dicot root. The outermost layer of dicot root is epiblemma. Many of the cells of epiblemma protrude in the form of multicellular root cells. False. Cortex has several layers of thick walled parenchyma cell with intercellular spaces. False. Innermost layer of cortex is called endodermis. Endodermis comprises of single or double layer of dash shaped cell with or without any intercellular spaces. Single layer of barrel shaped cell without any intercellular spaces. Endodermis comprises of a single layer of barrel-shaped cell without any intercellular spaces. Single barrel without any intercellular spaces. Dash as well as dash walls of endodermal cells have deposition of water permeable or water impermeable waxy material. Radial and tangential walls of the endodermal cells have deposition of water impermeable waxy material. Radial and tangential water impermeable. Radial and tangential water impermeable. Casparian strip is made up of that waxy material called suberin. Pericycle is thin-walled and parenchymatous. False. Pith of dicot root is large or small. Small. Initiation of lateral roots and vascular cambium during secondary growth takes place in pericycle. Parenchymatous cells between xylem and phloem is called conjunctive tissue. How many xylem and phloem patches are there in dicot root? 2 to 4. Still constitute all tissue in innermost side of endodermis such as pericycle, vascular bundles and pith. No endodermis. All tissue in innermost side of endodermis such as pericycle, vascular bundles and pith, but not the endodermis. All tissues innermost side of the endodermis such as pericycle, vascular bundles and pith. No endodermis. Monocot root. Monocot root usually have more than dash xylem bundles. This is called as more than 6 and this is called polyarch. More than 6 and this is called polyarch. More than 6 that is called polyarch. Pith is large in monocot root. True. Monocot root undergoes secondary growth. False. Identify the tissue given in diagram 9, able A to S. Diagram 9 is dicot stem. A. Epidermal cell, epidermal hair. B. Epidermis. C. Hypodermis. B. Epidermis. C. Hypodermis. And D. Parenchyma. E. Endodermis. F. Pericycle. G. Vascular bundle. H. Medullary rays. I. Pith. J. Epidermis. K. Collenchyma. L. Parenchyma. M. Endodermis. N. Pericycle. O. Phloem. P. Cambium. Q. Metaxylem or protoxylem and S. Pith. Dicot stem. A. Epidermal hair. B. Epidermis. C. Hypodermis. D. Parenchyma. E. Endodermis. F. Pericycle. G. Vascular bundle. H. Medullary rays. I. Pith. J. Epidermis. K. Collenchyma. L. Parenchyma. M. Endodermis. N. Pericycle. O. Phloem. P. Cambium. Q. Metaxylem or protoxylem. S. Pith. 121. Identify the tissue given in diagram 10, label A to J. Monocot stem. A. Endodermis. B. Hypodermis. C. Vascular bundles. D. ground tissue. E. Endodermis. F. Hypodermis. G. Vascular bundles. H. Phloem. I. Xylem. J. ground tissue. Diagram 10 is monocot stem. A is endodermis. B. Hypodermis. C. Vascular bundles. D. ground tissue. E. Endodermis. F. Hypodermis. G. Vascular bundles. H. Phloem. I. Xylem and J. ground tissue. Dash is the outermost protective layer of the dicot stem. Epidermis. Dicot stems are covered by thin layer of dash and bears. Thin layer of cuticle and that bears trachome. Thin layer of cuticle that bears trachome. Stems cannot have stomata on their wall. False. Cells arranged in multiple layers between epidermis and pericycle constitute the cortex. True. 3 zones of cortex are hypodermis, cortical layer, endodermis. Hypodermis, cortical layer, endodermis. Hypodermis is parenchymatous, collenchymatous or sclerenchymatous in nature. Hypodermis is colonchimatous. 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 Function of hypodermis: provide mechanical strength to stem. Provide mechanical strength to stem. Provide mechanical strength to stem. Hypodermis is a single layer. False. 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 Cortical layer consists of rounded, thick or thin-walled dash cells. Thin parenchymatous. Thin parenchymatous. Thin parenchymatous. The cells of endodermis in dicot stem are rich in starch. 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 Endodermis of dicot stem is also called starch sheath. Starch sheath. Starch sheath. Dash is present in the form of few semilunar patches of parenchyma or sclerenchyma on the inner side of endodermis. Pericycle sclerenchyma. Pericycle sclerenchyma. Pericycle sclerenchyma. In between vascular bundles, there are few layers of radially placed parenchymatous cells called medullary ray. Medullary ray. Medullary ray. Arrangement of dash in a form of ring is the characteristic of dicot stem. Vascular bundles. Vascular bundles. Vascular bundles. Pith in dicot stem is parenchymatous. True. 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 Pith of dicot stem is compactly packed with no intercellular space. False. Have large intercellular space. False. Have large intercellular space. False. Have large intercellular space. Hypodermis of monocot stem is colon or sclerenchymatous. 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 Each vascular bundle is surrounded by dash, which is dash chymatous. Bundle sheath, that is sclerenchymatous. Bundle sheath, that is sclerenchymatous. Bundle sheath, that is sclerenchymatous. Peripheral vascular bundles are generally larger than central ones. False. Smaller than central ones. False. Smaller than central ones. False. Smaller than central ones. Phloem parenchyma is absent. True. 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 Dash containing cavities are present in vascular bundles of monocot stem. Water. 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 Identify the tissue given in diagram level, label A to G. Diagram level is monocot leaf, A. Adaxial epidermis, B. xylem, C. mesophyll, D. substomatal cavity. Diagram level is monocot leaf, A. Adaxial epidermis, X. B. xylem, C. mesophyll, D. substomatal cavity. Monocot leaf, A. Adaxial epidermis, B. xylem, C. mesophyll, D. substomatal cavity. Identify the tissue given in diagram 12. Label A to J. Diagram 12 is dicot leaf, A. bundle sheath, B. xylem, C. phloem, D. adaxial epidermis. Diagram 12 is dicot leaf, A. bundle sheath, B. xylem, C. phloem, D. adaxial epidermis. Dicot leaf, A. bundle sheath, B. xylem, C. phloem, D. adaxial epidermis. Dicot leaf. Dicot leaf is dorsiventral or isobilateral. Dorsiventral. 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 Adaxial epidermis means which covers the lower surface. False. Upper surface. False. Upper surface. False. Upper surface. Abaxial epidermis bear more stomata than adaxial epidermis. True. 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 However, the amount of stomata on adaxial epidermis can never be zero. False. Adaxial epidermis can lack stomata. False. Adaxial epidermis can lack stomata. False. Adaxial epidermis can lack stomata. Leaf epidermis doesn't have cuticle. False. 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 Mesophyll is made up of dash chyma. Parenchyma. 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 Mesophyll have two kinds of cells. Name them. Spongy and palisade. Spongy and palisade. Spongy and palisade. Palisade parenchyma is abaxially or adaxially placed. Adaxially placed. Adaxially placed. Adaxially placed. They are arranged horizontally or vertically. 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 Palisade cells are parallel to each other. True. 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 There are no spaces between the spongy parenchyma. False. 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 In dicot or monocot, veins vary in thickness. In dicot, because of reticular venation. In dicot, because of reticular venation. In dicot, because of reticular venation. The vascular bundles are surrounded by a layer of thin or thick walled bundle sheath cells. Thick. Monocot leaf. Why leaf is called isobilateral? Because both the surface of leaf are same and equally green. Because both the surface of leaf are same and equally green. Because both the surface of leaf are same and equally green. Abaxial surface has more stomata than adaxial surface. False. Has equal amount of stomata. False. Has equal amount of stomata. False. Has equal amount of stomata. In monocot leaf, mesophyll is not differentiated. True. In dash, certain abaxial or adaxial epidermal cells along the veins modify themselves into bulliform cells. In grasses, certain adaxial epidermal cells along the veins modify themselves into bulliform cells. In grasses, certain adaxial epidermal cells along the veins modify themselves into bulliform cells. These cells are large or small, empty or filled, and colorful or colorless. Large, empty, colorless. Large, empty, and colorless. Large, empty, colorless. Function of bulliform cell. Curl leaf inwards, minimize water loss. Curl leaf inwards, minimize water loss. Curl leaf inwards, minimize water loss.